All right, well, 12 o'clock Eastern time, so let's get started. Thank you everybody for joining our webinar here today. The Canadian Public Health Association's Infectious Disease and Climate Change Webinar Series is exploring current and emerging infectious disease and climate change topics to share knowledge, research, and best practices. Before we begin, I want to acknowledge that the Canadian Public Health Association's office is situated on the ancestral and unceded territory of the Algonquin and Anishinaabe people. They have been the guardians of this land for millennia, and we are grateful for the example their stewardship provides. CPHA is committed to working with all First Nations, Inuit, and Métis people and their governments in realizing meaningful truth and reconciliation. Today's webinar, The Impact of Lethal Rat Control Techniques on the Ecology of Climate-Driven Vector-Borne Zoonotic Pathogens with a focus on Bartonella, is brought to you by the Canadian Public Health Association through the Infectious Disease and Climate Change Project. I'm Jillian Pritchard, Project Officer with the CPHA, and I'll be the moderator for today's session. First, we'll start with just a bit of housekeeping. It should be noted that all of our Zoom attendees are muted and their videos are turned off. But if you have any questions for our presenters, please type them into the Q&A box below, and we will moderate the questions at the end of the presentation. If you're having any technical issues, please also type them into the Q&A box and we'll respond to them. And if multiple people are experiencing the same issue, we'll respond to them live. We really strongly encourage attendee participation, and we really look forward to hearing all of your thoughts and questions. Just a few quick notes before we begin is that our webinar is being recorded and will be made available on CPHA's YouTube channel a few days after this event. And the presentation slides will also be shared in a follow-up email with all attendees. We would love to get your feedback, so please fill out our 5-minute survey at the end of the webinar upon closing the webinar window. And finally, the CPHA are holding an Infectious Disease and Climate Change Forum on October 5th of this year, which we hope you could all attend. So please visit our website if you're looking for more details. Now I'd like to introduce our speaker for today's webinar, Dr. Janet Hill from the University of Saskatchewan. She's a professor and head of the Department of Veterinary Microbiology at the University of Saskatchewan, Editor-in-Chief of ISME Communications, and past president of the Canadian Society of Microbiologists. She was born in Toronto and received her BSCH in biology and PhD in microbiology at Queen's University in Kingston, followed by a postdoctoral training at the University of Illinois and the National Research Council. Her graduate and postdoctoral studies include insect and plant virology, human parasitology, molecular diagnostics, and bioinformatics. Dr. Hill's research interests are in microbial diversity and ecology, and the role of complex microbial communities in animal and human health. Dr. Hill is a developer of DNA barcoding approaches based on the universal CPN60 gene, and her lab is home of CPNDB, a public database of chaperonin sequences. Dr. Hill is an enthusiastic teacher and research mentor of undergraduate and graduate students. When she isn't in the lab, she's usually watching and photographing birds. With that, I'd like to turn it over to our speaker, Dr. Hill.
Great, thanks very much, Jillian. Maybe I'll just share my screen here. There, how does that look?
Looks great.
Great. Okay, so thanks so much for the introduction. I really appreciate it. And thanks also for the invitation to come and speak to this group today. As Jillian mentioned, I'm a microbiologist primarily, and so this project was a really interesting opportunity to kind of learn about some other aspects of disease ecology and impacts of climate change. So I'll just dive right in here. What I want to do is start with some acknowledgements. The work that I'm going to talk to you about today was funded through the Public Health Agency of Canada, the Infectious Disease and Climate Change Fund. And it was a collaborative project between my team here at the University of Saskatchewan and also a group of researchers at the University of British Columbia, some of whom may be on the line today. We'll find out, I guess, because if I get any hard questions, I can turn it over. It's been really interesting. We had engagement from the Canadian Wildlife Health Cooperative and the Vancouver Rat Project that provided the initial samples and study system to make this project possible. So thanks to all those people, and I think you'll appreciate as we go through the story this morning why it was necessary to have such a team. So I'm sure it is, not something I need to explain to this audience, that we know that climate change is affecting vector-borne disease transmission and spread, and that these are ongoing effects. And so there's increasing amounts of interest in prevention and control of vector-borne diseases in this environment. And this was a quote from a recent review that was published in Nature Immunology, kind of gives a nice summary of some of the challenges. And they used the classic example of the Lyme disease and the Lyme disease tick. And it just gives a really nice encapsulation of the problem with vector-borne pathogens, where you don't just have the effects of climate change on the host or the pathogen, but you also have a vector involved and all the complications that brings. So if you're going to understand the effects of climate change on these sorts of infectious diseases, you really do need to have a very broad understanding of the systems that they live in. So the focus today is on rat-associated zoonoses and climate change. And so urban rats are known to be a source of zoonotic pathogens around the world. And we also know that humans are increasingly urban, more and more people moving into urban environments. And that means more rats, more human contact, and more opportunities for transmission. So climate change, as I just mentioned, is anticipated to drive changes in emergence and spread and frequency of all infectious diseases. And in particular, today we're focusing on rat-associated diseases. So we know one thing for sure, and that's that history has taught us that, the automatic response of just, well, let's control the reservoirs of these pathogens, that the results of doing that can be complicated, if not unpredictable. And there's some spectacular examples in the literature. Often people point to the example of badger culling and to control bovine tuberculosis, and the fruit bats and Marburg virus interactions. And in both these cases and in others, it's been shown that sometimes control methods that are focused on killing reservoir hosts have actually resulted in increased disease spillover to humans and domestic animals. And if not increases, then even just to say unpredictable consequences because of how complex these systems are. So it really puts the onus on us to step back and think about these pathogen host vector interactions with a more holistic approach. And so I want to just give you a little bit of background information that was really important to starting up the study I want to talk to you about today. And this was information coming from the Vancouver Rat Project. So this organization, this project was established 10 years ago and really dedicated to this concept of looking at the whole picture. So rather than just focusing on rats and pathogenic organisms or zoonoses, instead of taking the approach of looking at everything from rat health to their ecology and behavior, their impact on the people that live around them, and also how all of these things can influence thinking about management of rat infestations in the city. So the work that was really critical that had been done in the past for this particular project was some work that had been done over the last 10 years to show that rats in the downtown Eastside neighborhood of Vancouver were infected with a variety of zoonotic pathogens. And most of that work has all been published out there if you're interested in learning about some of the other pathogens that were detected. And it was also clear from a previous study that lethal interventions, so kill trapping of rats, changed the pattern in some cases of pathogen distribution in those populations. And the example I wanted to point out to you was that what they discovered was that the odds of colonization with Leptospira interrogans were actually 10 times higher post-intervention than before intervention, which is a bit of an unexpected result. So the one way of explaining that would be that by killing rats, they live in a very socially organized colonies, and so killing rats disrupts these colonies, it disrupts social hierarchies. That's going to have an impact on their behavior and maybe change patterns of their contacts and the number of contacts they're having, and potentially increase opportunities for transmission of an agent like Leptospira interrogans. Of course, the interesting thing is that that is a directly transmitted pathogen. So the question that sort of inspired us to seek the funding to do this project in the first place was, well, what about vector-borne pathogens? Would we anticipate that if you looked at the same sort of system, but instead looked at the prevalence and the burden of a vector-borne pathogen in this environment, would it look the same or would it look different? So other groundwork fortunately pointed us in the direction we needed, which was in fact to a vector-borne pathogen, which is Bartonella tribacorum. So back in 2011, 2012, there was a study done by the Vancouver Rat Project in the downtown Eastside neighborhood that showed that Bartonella tribacorum was in fact endemic in the rats in that neighborhood. And in fact, about 25% of over 400 rats that they tested were positive for Bartonella. And a subsequent survey of some of the people living in the neighborhood showed that there was serological evidence anyway of human exposure to this organism. So this organism, Bartonella tribacorum, falls into a genus of bacteria, Bartonella. These are transmitted by blood-sucking arthropods. So that includes ticks and fleas and lice. They're facultative intracellular organisms. They live part of their lives inside cells. Sometimes you'll see them referred to as a stealth pathogen, and that's because of their habit of hiding out inside red blood cells. There have been at least 67 species of Bartonella identified and defined. About 10 of those have been associated in some way with disease in humans. You may be most familiar with Bartonella henselae, which is the agent of cat scratch disease. And then there's Bartonella quintana, which was famously associated with trench fever during World War I and is transmitted by lice. So they have a widespread distribution, and that's in both terrestrial and aquatic mammals all around the world. So something like 50% of feral cats and wild rodents and 90% of wild ruminants have been, in various studies, have been reported to carry some Bartonella species. So how the life cycle works in general, in the case of Bartonella tribacorum, we're talking about something that's transmitted by fleas. So fleas on the surface of the skin will defecate. The organisms can make their way into the skin where they can infect cells within the dermis and then eventually end up in the endothelial cells of the vascular system. So once they're in there, they have access to red blood cells. And like I said, they have a habit of hiding out and multiplying inside erythrocytes, which means they also have the opportunity to be taken up again by another blood-sucking vector. So they have a very sneaky strategy and they're quite successful at being transmitted. So Bartonella in rats, in particular genus Rattus rats, they've been reported around the world, and this is a figure from a fairly recent paper just summarizing the species of Bartonella that have been detected in Rattus rats around the world. And this blue dot here is the Vancouver Rat Project study reporting Bartonella tribacorum, but you can see there's plenty of other reports around the world. So the other thing besides the opportunity that we knew there was a vector transmitted pathogen in this population, we also had access to a fantastic collection of samples that had been put together from this neighborhood. So what you're looking at here is an aerial view of the downtown Eastside neighborhood in Vancouver. And it was all these various shapes here. The blue and the orange rectangles are the various sites that were outlined. Each one consists of 3 city blocks. And within each block in the back alley, there were 10 live traps put out for rats. And then these blocks were assigned to either as a control site or an intervention site. And the orange ones are the intervention sites. And what you can see is the center block in each one of these intervention sites was where culling actually took place during the intervention period. So the way that the samples we used in this study were from this particular workflow here. So after some pre-baiting, there was a 2-week period of capture and release during which rat blood was collected and fleas were combed off or shaken off the rats and collected as well. So then after this initial sample was taken, there was a period of intervention, and that was either in the control blocks, just continuing capture and release, or in the treatment blocks, in the center blocks of these intervention sites, there was cull trapping that went on and rats were removed from those blocks. So then after another period of time, there was another period of capture and release. And again, rat blood and fleas were collected for comparison to these samples back here. So the other challenge we had in setting up this study was, well, how are we going to detect Bartonella? Previous evidence had showed it was present in the population. In those previous studies, the method that had been used was by culture. So blood was actually cultivated on agar plates and based on colony morphology and selection in the media, presumptive identification was made as Bartonella. And then those colonies were further identified by PCR and sequencing of a gene that encodes a protein called citrate synthase. So the challenge with this approach is that it can take up to a month to produce colonies on media with Bartonella. They are tricky to grow. These culture-based methods are a little bit limited as well in terms of the need to have samples that are very carefully preserved before they are cultivated so that you can ensure organisms are viable. In the case of mixtures of infections, it can be a real problem because even if you have many, many colonies on the plate, you're not going to identify all of them. You're going to pick something that's typical of the morphology you see and move on from there. So we put our heads together and thought that, well, maybe it would be a better approach to take a more molecular diagnostic approach to this. So that sort of set us up for the main objectives for this study. So the first of which was to establish the molecular diagnostic methods. And in this case, it was real-time polymerase chain reaction, or PCR, that we did. That was to detect and identify Bartonella in rat blood and fleas. The method had to work on both. And then we were going to apply those methods to, number one, to determine what species of Bartonella were present in the samples that were collected through 2016 and 2017. And then use the experimental design that I just showed you on the previous slide to investigate the effects of that lethal rat control on the prevalence of Bartonella in the rats and their fleas. Okay, so just to recap there, so sampling happened over the summer and into the winter of 2016-17. The rats were collected from these 36 city blocks, either from control or intervention sites, and blood and fleas were collected. In the case of the fleas, from 1 to 5 fleas per rat were actually put into a pool. So you'll hear me refer to rat pools, and I don't mean a little swimming pool. I actually mean a collection of rats that are put together. And then we only looked at the pre and post intervention samples. So in total, we had blood from 480 rat blood samples, and all the rats were Norway rats. And we had 200 flea pools, and all of the fleas that were identified were northern rat fleas. So just won't get into the methods in huge detail, but I just wanted to give you a feel for this. So for the rat blood samples, essentially we purify total DNA from the rat blood, which of course will include rat DNA plus any microbes that are there. We perform a PCR for cytochrome B, which is just a housekeeping gene that allows us to make sure the samples are okay. And then we use this genus-specific PCR technique to detect these GLTA genes. And by genus-specific, I mean this assay should amplify the GLTA gene from any species within the genus Bartonella. So we're not biasing, hopefully, toward one species or another. It's an endpoint analysis, so the result is positive, negative. We're not doing quantification here. And then if we got PCR products, if we got positives, then we sequence them in order to figure out which species, because remember, the assay can amplify any species within that genus. So for the flea pools, they were surface sterilized and then DNA was purified from them. We detected a housekeeping gene belonging to the flea, again, just to make sure that the samples were okay. And then we started out using the same assay detecting these GLTA genes for Bartonella genus-specific detection. We had a technical issue with some of the batches of samples that we were processing, which actually turned out in retrospect to be a good thing because it taught us something important about some of these diagnostic tests. So we switched to a different assay, and this targets a gene called SSRA. It's also supposed to be a genus-specific assay. And again, an endpoint analysis. So we're just getting a positive, negative reaction. And we can identify what species if we sequence the product that's made. So we include a boatload of controls with all of this for reasons that any of you who work in the lab know are super important to try and rule out the possibility that any positives you detect are not false positives. So what we found with the rats was pretty straightforward. So we screened 480 rat blood samples, and about 20% of them were positive for GLTA, indicating that some species of Bartonella is present. Interestingly, though, when we sequenced them, we found that it was about a 50-50 mixture between Bartonella tribacorum, which had been previously reported in this population, and another species of Bartonella called Bartonella vinconii, which had not been previously reported. So that was super interesting right off the start and opened up a whole bunch more questions. Another thing was that at least one of those samples appeared to kind of be giving us a bit of a mixed signal and got us thinking about the possibility of multi-species infections. When it came to the fleas, we started out screening with this GLTA assay, and we're getting about 48% positive, which isn't surprising because they're pools of multiple fleas together, they're not individuals. And then of course we had to switch. So we ended up screening the whole set of samples with this different assay. And about 42% of those were positive. But interestingly, when we sequenced them, they were all tribacorum. There was no vinconii detected using this SSRA assay in the flea pools. So we came away from this with two big questions. First of all, are these two assays detecting both species equally well? Because it looks weird. And the other thing was, are mixed infections a thing here? Are we actually dealing with multiple species of Bartonella? So we did a little bit, you'll bear with me with this one kind of geeky slide about some of the troubleshooting we did, but we basically took these 144 flea pools that we had screened with both assays and compared the results. And what we found was in most cases, the results were concordant. We got a positive or a negative in both cases. But what was interesting in the ones that were not concordant, the skew was definitely towards samples where the GLTA assay was giving us a positive, saying, hey, there's Bartonella present in this flea pool, and the SSRAs were negative. And again, if we looked at the GLTA products, we were able to detect both species. So we're starting to think, well, maybe the SSRA assay is not actually detecting everything it should. So we took advantage of the fact that we had all these rat blood samples, and we just selected randomly some negatives, one rat bloods that were positive for tribacorum, and some that were positive for vinconi. And then we tested them with the second assay. And what you can see is we got great agreement on negative, and we got really good agreement on the tribacorums. But what happened was that the vast majority of the vinconi positive samples, the SSRA assay just doesn't detect them. And that's important because this assay has been out there for a long time. It's been applied in a lot of studies. And it does seem, at least in our hands, that it doesn't do everything that we had hoped it would do in terms of detecting all the species of Bartonella. So the mixed infection story was another one that was really interesting. And if you just sort of imagine you've got a sample that has a mixture of blue and red bugs in it, and you're trying to describe what's there. If you do culture, a lot of the time your conclusion from this would just be, well, it's blue stuff. And the reason for that is really a question of numbers. So again, if you grow, you got 100 colonies on a plate and you pick one or two of them to identify, chances are it's going to be the most abundant thing that's there. So you may miss a mixture where there's a big skew in abundance. If you do conventional PCR methods, essentially what you're doing from this whole sample, and you're taking all the DNA out of here, so you're producing a pool of pieces of DNA, amplified pieces of DNA. And obviously the more abundant thing in the sample is going to be overrepresented in that pool. And if you do conventional or what's called Sanger sequencing on that, what you get is essentially an average of these sequences. And so that result is going to look an awful lot just like the blue thing. And you're not going to see the signal from this less abundant sequence. So the solution that we thought we would apply based on experience with other systems was to take advantage of next generation sequencing. And these are a whole set of techniques, various brand names and various technologies and chemistries, but the basic idea here is that you are actually, rather than sequencing a pool of products and getting an average, you're actually sequencing individual molecules. So in the technology that we used, these molecules are actually arrayed onto what's called a flow cell, and then each individual molecule, the sequence of that is determined and reported. So even if these red things are quite rare, when you look at the number of sequence reads that you get out of the machine, which is thousands and thousands and thousands of different results, you will in fact see that rarer sequence represented. So simultaneously, we can look for all sorts of different things at the same time. So that's the approach that we took. So we took 94 samples that were positive for either GLTA or SSRA in our PCRs, and we generated a lot of data. So we ended up generating about 375,000 individual sequences, about 4,000 per sample. And the important thing here is that our negative controls don't give any meaningful data. So that means we don't have background contamination. And what was interesting was when we take all of that data, those 375,000 sequences, all boiled down to essentially 4 unique sequences, or variants as we'd call them. And those accounted for something like 98 or 99% of the data. 2 of those sequences looked like tribacorum and 2 of them looked like vinceni. So we can in fact detect both species. And what was interesting was if we look at the distribution of them, what happens is we have, definitely a big chunk of samples that are positive only for tribacorum. We have a smaller number where all we detected was vinceni. And then we have quite a large proportion of our samples that in fact had both present. So it looks like mixed infections are actually fairly common and both of these organisms are present. So in terms of the vinceni thing was really interesting because we hadn't detected it before. We did a little bit of phylogenetic analysis. So this is essentially a family tree of Bartonellas based on that GLTA sequence. So down here in this sort of green block, this red label here represents our most common study sequence from this work. And then this second line here, this uncultured Bartonella, is actually the sequence from the 2012 study in this neighborhood by the Vancouver Rat Project. So it's exactly the same sequence. And then up here in this larger blue box, you see 2 subspecies of Bartonella vinceni. There's something called subspecies burkhoffii, and there's something called subspecies vinsonii. And our study sequences fit into this cluster with the subspecies burkhoffii sequences. We did a little bit more work on this, looked at another gene in the genome of Bartonella, and discovered that we could identify it further as Bartonella vinsonii subspecies burkhoffii type 2. It's a very specific identification. And what's interesting about that is that this organism was first described actually as recently as 1996 in a dog. And it has been associated with endocarditis in humans as well. And it's known to have quite a few wild carnivore reservoirs, including mostly reported as coyotes, but also foxes, skunks, and raccoons. So it's kind of interesting to think about the other dynamics that might be going on with other hosts for this organism in Vancouver and in Canada, right, frankly. So the other important thing is that we didn't detect it. It was not detected in the previous study in the same neighborhood. And in fact, it hasn't previously been reported in Rattus rats. So that was quite a novel finding. I do wonder, of course, as I'm sure you are wondering, is, well, why is that? So if we think about it, well, maybe Bartonella vinsonii is a new arrival since the 2012 study, or maybe it was there, but it wasn't detected. So a couple of things we did was we wondered, well, if the vinsonii samples were all spatially clustered together in the neighborhood, and that particular area just happened to not be sampled in the 2011-12 study, that would explain things. But in fact, there was no spatial clustering of the tribacorum, the vinsonii samples, or the mixed infections. And here in this diagram of the neighborhood, you can see the green boxes actually are showing you the 2011-12 study area. So really good overlap too. So it seems to me more likely that maybe the difference here, well, either it is a new arrival, or the method of detection made the difference, and that by using a culture-based approach, this less abundant organism might not have been detected. So most of this part of the study is now published. If you're interested, in Frontiers in Veterinary Science came out fairly recently, so you can read the gory details of the technical aspects of the study. But I wanted to get back to just for a minute here, our third objective, because this was kind of the big culmination is, okay, great. So you've detected 2 species of Bartonella. What's the effect of the lethal trapping? So again, just as a reminder, we had samples from before the intervention and after the intervention and wondered about how that culling affected. So Kaylee Byers and her team in Vancouver did a whole bunch of fancy statistical modeling that I have no hope of explaining to you whatsoever. But what I can tell you is the conclusion. And the conclusion from this was, interestingly, that the carriage of Bartonella was significantly more likely among rats caught after the intervention in the control and the flanking blocks, but not in the intervention blocks. So you anticipate an increase over the study period going from summer to winter. That's been established before. And that didn't happen in the blocks where there was culling. And also another thing that came out of this was that there was no association between the intervention and the number of fleas per rat or the carriage of Bartonella by fleas. So it didn't seem to affect the fleas at all. So the intervention seems to have prevented this anticipated seasonal increase in Bartonella. But how, right? And I don't have a perfect answer for you, but what I do have is a couple of thoughts on it that we'd sort of considered, and perhaps we can discuss this a little bit more as I wrap up here. So reducing the rat density in an area could have a lot of knock-on effects. I think that you need to consider the effects on the rats themselves in terms of their behavior and the types of interactions that they're having with each other. Rats live in communities, and their burrows tend to include nests that are shared by multiple animals. And so if you reduce the density of rats in a particular area, maybe there's less nest sharing going on. And another really important thing is that these fleas are thought to spend an awful lot of time, in fact, maybe more time in the nest than they do on the rats. So if you remove rats and the fleas that are associated with them from the community, in fact, you may not be making a dent at all in the flea population. And so that's another thing that really needs to be considered, is the distribution of those vectors on their host and in the environment around. So it is possible that reducing the rat density has decreased the interactions or their exposure to each other's fleas, or the number of rats that a particular flea might be on in its lifetime. So all of those things are interacting with each other, no doubt, to explain this. But I think the take-home message here is really more about just how complicated these systems are. And this is yet another piece of evidence in that direction. So what we took away from this was, number one, that rats and fleas can be infected with more than one species of Bartonella simultaneously, that Norway rats and northern rat fleas may be hosts for Bartonella vinsonii subspecies Burkhoffii, which has not been reported previously, that metabarcode sequencing or next-gen sequencing is a viable option, especially for detecting mixed infections of related species, and in fact offers a lot of advantages over other more targeted methods. So something definitely to consider for future studies. And then finally, removing rats may prevent increases in Bartonella prevalence, which potentially could lower human health risks. But I think we have to keep in mind that on the flip side of that is the observations that were made with Leptospira that had the opposite effect. So it is a complicated situation for sure. So that's all I have for you. Just again, to turn back to thanking all the collaborators who worked so hard on this project in getting this work done, and the folks who donated samples and controls to us, and of course our funding from the Public Health Agency of Canada. So with that, I will stop talking, and if there are any questions or thoughts, I'd love to hear them.
Thank you so much, Dr. Hill. That was really interesting. It's neat to see what is being done with the Vancouver Rat Project. Yeah, so if any of our attendees have any questions, you can type them into the Q&A box below, and I will read them out for Dr. Hill. I'll just share my screen here. Okay, well, I'm not seeing any questions yet, so we'll give people a minute to type them in, but I'd love to start us off. But this was really interesting work in Vancouver, but is there more work going on across Canada to understand how Bartonella is affected in rat populations elsewhere in the country?
Yeah, not that I'm particularly aware of. I think it's kind of important to point out what a special kind of project, and I can say this is not a member of the Vancouver Rat Project, but this has been a really fascinating project. And I think the kinds of information they're getting out are just so interesting. And it really is like urban wildlife in general is something that I think we're finally starting to appreciate how important it is. I know in Ontario, there's been lots of work thinking about rabies and other potential zoonotic pathogens there as well. But we, as we encroach on wild places, we see more and more wildlife in our cities, and which is a good thing or a bad thing. Some of you may be following all the drama about echinococcus in Western Canada appearing more and more in coyotes and dogs there. So yeah, anyway, certainly an area ripe for study, because most cities, except for of course Alberta, have rats.
All right, we've got our first question here from an attendee asking, did the nature of the rat's nest habitats change over the study area?
Yeah, that's a question I can't answer. And I don't know actually if anybody from the Vancouver Rat Project is on the call, feel free to jump in and show yourself. But it's a really interesting question, I think. And I don't know a lot about that, but it would be fascinating to know whether or not, in addition to just the movement of the animals changed, that they actually renovated. Yeah, I don't know.
Okay. And what's the difference between the Bartonella species and the mystomys found in Africa?
I don't know.
Okay.
Do you?
No, I do not.
Does anyone?
Well, we can put that on the
I don't know.
burner if anybody wants to jump in with that.
Sure.
They can. Okay, we've got another one here. You mentioned that Bartonella vinsoni has been identified in coyotes, red foxes, skunks, and raccoons, all somewhat urban animals. Is it believed that these animals pass this pathogen to the Norway rats via the fleas? And also, capture and release, does this mean releasing the rats again in the same block? And if so, how are the permissions obtained from the local residents?
Yeah, so with respect to the potential urban reservoirs like coyotes and foxes and stuff, I think that's a really fascinating question, and I think it's a question that we don't know the answer to, but might be something worth investigating. I mean, it's certainly feasible, depending on the opportunities for contact and sharing of vectors. But I know that coyotes in particular have really become quite common in the city, and I think it's a reasonable hypothesis, but remains to be tested. With respect to the capture and release, yes, they were released back from where they were found. And all of these, the studies were, of course, went through all the ethics approvals that they had through the university and working with the community as well. This is if you're interested, there's actually some really neat work that's been done, and a lot of it's available on their website, about work that the VRP has been doing in terms of looking at impacts of rats on people in the neighborhood, because it's something that, again, I think was not particularly well appreciated before, about the the psychological effects of living with rats. And there's some really interesting stories there that I would encourage you to look at.
I'd love to get a little more into the human health impacts. Was those looked at, or do these two species of Bartonella have any direct human health impacts?
Yeah, so tribacorum, not such great evidence, but Bartonella vinsoni, there definitely have been reports of, I think in particular, like endocarditis, but the Bartonella infections are really challenging in the sense that I think the symptoms and how they present can be extremely variable. So a lot of the time, the descriptions of the symptoms are very vague, and they're not consistent from person to person or species to species. I suspect it's a pathogen that kind of goes under the radar a bit. So I think there's still quite a lot to learn there. I think we know more about, say, cat scratch disease and the trench fever type infections, but some of these others, I think we probably have a lot to learn about those.
We know that climate change is really affecting vector-borne diseases of where we find them, how they spread, and their interactions with human health. Can you speak more to that on Bartonella specifically?
You mean in terms of the climate change effects?
Like, do we expect to see more in the future? Or?
Yeah, I would expect so, just in the sense that, like, if we, it depends on the system, right? Because, like I said, there's 67 species, and I'm sure more to be discovered. But I think that the urbanization and urban population density issue, if you think about living conditions and the presence of these sorts of vectors, so with increasing urbanization and crowding, and if you have poor living conditions and issues with the presence of the vector, I anticipate that's the case. Warming temperatures as well may affect the survival of the fleas and their and the reservoirs in the environment, for sure. It's interesting, actually, we have some researchers at the University of Saskatchewan who are working on looking at Bartonella in the Arctic and nest fleas of geese and other Arctic animals. And there's another environment where we might learn quite a lot, because the climate change effects there have already been so much more dramatic.
Oh, it's very interesting. We've got lots of questions coming in. One asking, what are your recommendations to local public health agencies from what you learned in this study?
Well, I would not be in a position to make recommendations to public health agencies, but what I will say, though, I think, is that one thing that really came out of this study for me was an appreciation of just how complex these sorts of systems are. And that really there's no other way to look at this except for to take that ecological approach and to really look at all the components. Because I think part of the problem is, like, there's all kinds of rat control going on by individual property owners and everything else. But in terms of what the wider impacts are of that, I don't think we know. And I think it's just that it's very difficult to predict, I think, what will happen. I think the more cases that we see and the more studies we do like this of specific pathogens, we start to get a feel for what the menu of options are. But I think it still remains very difficult to predict. So I think caution is really important, because the effect of what you do may in fact be quite different from what was intended. But we're accumulating better and better tools, and I think this kind of work is just really important to inform those decisions. And it takes a village, right? Like it's a combination of having the right methodology and but the people also with the expertise in the ecological system and the biology of all the hosts and vectors involved, and the people involved as well. So that interdisciplinary approach, I think, is so important. It's just critical.
I think it was very evident in your presentation how much work and time and interdisciplinary expertise went into this project.
Yeah.
I've got another question. How is human behavior affecting the distribution of rats in the study area?
Yeah, that's an interesting question. I think, probably one of the, you think about, that could be in a number of ways, right? There are humans who are doing rat control, for sure. So that's probably affecting distribution of rats on a small scale. Density of population would definitely, you think about food sources, right? Handling of waste in the neighborhood and everything else is going to have an impact. I suspect traffic patterns, available nesting sites, construction, how well buildings are maintained, all of those things would be affecting the distribution of rats in that area for sure. They are survivors.
As you said, it's very complex.
Oh, there's the answer to our Lassa fever. Oh, okay.
Yes.
Yeah. Sorry, I will admit complete lack of knowledge on that topic, but I'm going to go look it up after this.
All right. And the person who asked the question about the difference between Lassa fever and Bartonella, feel free to follow up and we can maybe answer that question offline. Yeah. Okay. Another question here. On the map you showed, various strains of Bartonella were shown. What had the unexpected Vinsoni been found before? Or sorry, where had the unexpected Vinsoni been found before?
Yeah, so it wasn't found before. We only found it in the 2016-17 samples. I wish we could go back in time to the samples that were collected in that 2011-2012 study and re-examine them using the different methodologies. But unfortunately, I don't think that's possible because I would love to be able to answer that question. And as I pointed out, that species, Vinsonii, hasn't been reported anywhere in these rats. So
I don't see any more questions coming up, but I'll ask one last
one.
But where would you like to see this project or this work go in the future?
Yeah, I think it would be really interesting to branch out and look at some of the other agents that have been detected in these rats. So there's MRSA and Listeria and Bartonella and others. So that would be super interesting. I think that with the technologies that we have now, we could probably do a more comprehensive description of like viruses and bacteria and protozoa that are in this population. So as a lab person, I'd say that was interesting. And I think ongoing surveillance is also something that would be definitely worth pursuing, because I think we're going to understand, we have some sort of baseline data now, but to understand how things are changing over time is going to be really important. So, yeah. And I think, as you mentioned right off the top, that there's opportunities to do this kind of thing in other locations in Canada and beyond. And I think that would be really interesting to compare. So, do we see the same Bartonellas in Vancouver as we do in the Pacific Northwest in the US or in Eastern North America?
Yeah, absolutely. It sounds like there's it's a topic that's very ripe for more inquiry.
Yeah.
Another question come in. With the promotion of composting activities, including from local food vendors, as an eco-solution to waste disposal, would you expect the local rat population and associated disease to increase as well? What is the single most effective way of reducing disease spread from rats? So like effective containment of food waste.
Yeah, I mean, I would guess that's probably really important. And I don't know much in specific about what's going on in Vancouver in terms of like the composting activities and how that's set up. But I think there's a lot of thought needs to go into the design of those systems. I'm sure that it can work, but you definitely need to probably design with rats in mind. It's like the search for the raccoon-proof garbage can, right? I mean, it's a challenge for sure. And in terms of effective way of reducing disease spread from rats, I mean, it's all about exposure, right? You're not going to have disease transmission if there's no opportunity for exposure and transmission. So I think that there's some pretty significant issues around quality of housing, access to, to good quality housing that's safe and hygienic, and just reducing the exposure of people to the vectors and the animals that are the sources of these pathogens for sure.
That's a great note to end on. So thank you so much, Dr. Hill, for your presentation today. It was so interesting to learn more about this project. And thank you to all of our attendees for joining us today. Be sure to fill out our survey once you close the webinar window. It should only take 5 minutes, and we'd really value your feedback on this webinar and our Infectious Disease and Climate Change series. So thank you very much to you all, and take care and have a good day.
Okay, thanks everyone.